So just a quick introduction. Some of you know Sebastian, but Sebastian is the founder and CEO of PAIA Biotech, based in Cologne, Germany. And he has some novel assays for quantifying characteristics of antibodies. And today he is going to talk about some glycan properties, including afucosylation. And so I'm going to turn it over to Sebastian.
Thanks, Chris, and welcome to this webinar, everyone. Great that you can take the time today to listen to my talk about afucosylation. So this is going to be the focus of my talk, and how we use our special kind of assay technology to provide a means to screen early and very quickly a lot of samples in cell line development. So I would like to start with a very short introduction to those who don't know PAIA yet. So quick introduction on PAIA Biotech. As Chris mentioned already, we're a German company based in Cologne, and we do have a special kind of a platform technology that we've been patenting, and that allows us to simplify, to streamline bead-based assays. And with this kind of assay technology, which is centered around some quite fancy microplates, we do two main things. First is we have developed high throughput titer assays. Actually, a new version for the FC titer assay was launched last month, which is extremely fast and really nice, a new assay system. I'm not going to talk about that today, but we're going to focus on the assay kits that we've developed for glycan screenings of antibodies and FC fusions. So that not only means fucosylation, but also includes other features like galactosylation, mannosylation, Man5, big topic for antibodies, and of course silation, which is the most important feature if you're looking at FC fusion proteins. But again, focus will be afucosylation today. And we do have two partners that we work very closely with. The one is on the technology side, which is Genentech, a company which is also a German company, and they develop and market very nice cell imaging systems with fluorescence, which we need for reading our assays. And we're working in the US with Chris and his team from Techmatic. They're based in Illinois. So that much as an introduction. So let's get to afucosylation. And firstly, I quickly wanted to remind us why we do all this. And the reason is because afucosylation has a very strong impact on ADCC. And this sketch here shows you how this antibody-dependent cellular cytotoxicity works. So the therapeutic IgG is recognizing the target cell, often that's a cancer cell, binds to it, and then recruits by its FC part natural killer cell, NK cells, which then do their job and kill the target cell. That's a very common mode of action for drugs against cancer indications. And this effect can be strongly influenced by the fucosylation on the FC part of the IgG. And this was first published in 2002 and 2003 by two separate groups from Genentech and from Kyoahako in Japan. And it took, precisely 10 years until the first drug, which was afucosylated, was approved in 2012 in Japan. And since then, we've seen really an increasing number of clinical trials involving those afucosylated IgG, mainly in cancer. And that's what we also see at PAIA that there's an increasing need and demand for screenings for afucosylated antibodies. So when people characterize the different glycoforms, especially also the afucosylated glycoforms, in the context of ADCC, they're mainly looking at two different kinds of assays. One is the Fc gamma receptor 3A binding, which can be done, for example, in SPR systems, so surface plasmon resonance. That's what you see here on the left-hand side, multiple injections of increasing amounts of different very well-defined glycoforms. And you see that the different glycoforms bind with a different affinity towards those receptors. And I don't want to go too much into detail here, but the lower the degree of fucosylation, the higher the receptor binding becomes. And in this particular study, it was suggested that also galactosylation has a certain effect on this receptor binding. And then if the galactosylation is also kept with silylation, this affinity is diminished to a certain extent. So that's the one part or kind of functional assays that one does very often when it comes to testing these different glycoforms. And on the right-hand side, you see a typical ADCC assay where we're looking at the response of NK cells to antibodies, and these curves here show the effect of differently fucosylated antibodies. On the left-hand side is the IgG with the lowest amount of fucosylation. On the right-hand side is 100 fucosylated antibody, which is usually the case if you don't do any kind of special kind of engineering to your CHO cell line, for example. So that's a huge effect that you can obtain by reducing the amount of fucosylation. And what is interesting in this plot also is that, for example, this blue line is something like 20% remaining fucosylation, so 80% afucosylation. So you can already get quite a big effect on the potency of the drug, even if there's a significant amount of fucosylation left on the antibody. So this is the one thing that I want to highlight here. The other thing is that it's not only the natural killer cells which have an impact on the ADCC, but there are also other cells which play a role there. Polymononuclear cells are involved there, and they also tend to prefer fucosylated IgG. So it's not necessarily a complete to get the best ADCC. So that really suggests that it's desirable to be able to tune the fucosylation to a certain extent in your production process. So that much for afucosylation and ADCC. And let's look at how people produce afucosylated antibodies and what are the different approaches to get there. So what people do, of course, is cell line engineering, and that can involve genetic engineering or the addition of special supplements to the cell culture, or a combination of both. And that can either lead to complete or to partial afucosylation of the antibody product. And kind of the most straightforward approach to get there is by knocking out the fucosyl transferase here in the middle, which means that all the glycans which are formed usually in this pathway and which are normally fucosylated become afucosylated. So with this kind of method, you get to very, very low degrees of fucosylation with not a lot of ways of fine-tuning that. You can also knock out genes further upstream this process, but this is not so common. So the knocking out the fucosyl transferase here is the original approach that Kyowa was pursuing early on. Then we have other ways. For example, that's what Roche glycan is doing, overexpressing of these two enzymes to divert this pathway into this direction, which then leads to a production of bisecting glycans to quite a high degree, like 60–70%, and all lacking fucose. So these are two of the genetic engineering approaches. Another one would be, and that's what's shown here on the top, is knockout of these genes here, which kind of inhibits the cellular synthesis of fucose precursors here, so that the afucosylation cannot be done by the cell itself, but one has to feed in fucose from the cell culture media, and that is a means to kind of fine-tune or modulate the degree of fucosylation in the final product. So I think those are the most important methods for obtaining fucosylated antibodies at the moment. So what does that mean for cell line development? First of all, it means there can be quite some clonal variation in terms of afucosylation. That's what you can see here on the left-hand side in a plot. I think it's from Roche glycan. You see, I think this is almost 250 or 300 different clones with, as you can see, different degrees of afucosylation. So yeah, quite a large span between starting from something like maybe 30 up to 60% afucosylation. And as a kind of binote here, you see how nicely that correlates with the affinity towards the Fc gamma receptor in an HPLC setting. So quite some clonal variation there. And on the right-hand side, you see in an approach from Genentech, where they knocked out the Fc gene, the FX gene, that different kind of clones respond differently to fucose supplementation in the media. So the general trend is kind of the same, but if you look at the red bars here, you can see they respond kind of differently. So also in terms of media compositions, clones react differently. So it's certainly in both aspects desirable to have screening technology in place that allows you to see the trends early on and quickly, so you can select, for example, clones that have the desired fucosylation profile. So let's look at the assays that we have developed at PAIA for this purpose. And I want to start with one slide that I guess, several people in the audience have already seen, so sorry for that. So this is the principle of our PAIA PA201 assay. As I mentioned briefly in the introduction, it's a bead-based assay, and in this glycan assay, we use a sandwich format. So we have a capture bead that captures the IgG, and then you have the glycan sitting on the Fc part of the antibody, and then we use fluorescence labeled lectins to bind and detect these glycans. This assay involves a sample prep step with a 5-minute heating, which helps us to expose the glycans on the IgG. The typical IgG sample concentrations are sometimes, well, somewhere between 50 and 200 micrograms per mil. The assay time for the whole plate is something like 90 minutes, which means that if you run afucosylation only screening and you run each sample in triplicates, you're able to run 96 samples in such a setting. And here on the bottom is depicted the workflow. This is a well. This is the labeled lectin that binds to fucose. You add the sample. As I'll explain later, that is a diluted supernatant. It's mixed on an orbital shaker. Afterwards, the beads are settling to the bottom, and then with fluorescence bottom reading, you determine the amount of lectin that has bound to the fucose on the IgG. So the lectins, as I said, bind to the specific sugar residues on the glycans, and we get binding rates for the different lectins. So this is kind of a setup that we would use in other glycan assays where we have different kind of lectins for the different sugars, sialic acid, galactose, mannose, and fucose. So fucose is going to be the one that we would use for afucosylation screening process, obviously. And if we look at how specific this lectin is, one of course likes to first check whether enzymatic treatment of antibodies removes all the lectin binding, and we've done this here on this slide with two different kind of enzymes. First is PNG-ASF here on the left-hand side, and you see that all the lectins, or the binding of the lectins, is removed by that. So that's PNG-ASF, and on the right-hand side it's endo-S2 enzyme, which leaves a little bit of glycan on the antibody. So you have these glucosnax here, and the core fucose is also still on the protein, and you see that still the core fucose binding lectin does not bind anymore, or just a very little amount. So it seems that this core fucose binding lectin really needs also some of the N-glycan core structure for binding. So that's usually 3 more mannose sugars which are located somewhere here. So that's the one experiment that I wanted to show you. And the other thing is the dynamic range of the assay. And for this purpose, we have generated calibration curves. And here I want to show you 2 examples where we mixed fucosylated and afucosylated IgGs. On the left-hand side, it's really an afucosylated IgG mixed with a fucosylated, and on the right-hand side, it's a fucosylated, kind of very normal IgG with almost 100% fucosylation, and then a variant of the same IgG with no glycans at all. And in both scenarios, you see quite nice calibration curves with an almost linear range of like 100% afucosylation to maybe 30, 40, whatever. And then there's a tendency that this curve kind of flattens out here towards 100% fucosylation. So for this whole range here, that's going to be the assay is quite nicely able to determine the different degrees of fucosylation. But you will have a hard time to differentiate between 90% and 95% fucosylation. Okay, so how do we apply these assays in a CLD screening process? So I want to show you an example of how we approach this. So first of all, what are the goals for afucosylation screenings in CLD? First of all, of course, we want to be able to perform a fast screening of potentially hundreds of clones those samples should be non-purified, so diluted supernatants. And we would like to be able to rank the clones with regards to the fucosylation levels and then do selection based on that. And of course, what would be really desirable, and that's what we can do with our assays, is that the screening can be done very close to the process in the CLD group where the samples are generated. So what do we need in terms of equipment for this? Orbital shaker, the faster the better, because that means the faster the assay is done. Then for the sample prep, we can either use something like a thermocycler, but you can also use an orbital shaker with a heating function, which is reliable. And then the last part, of course, is a reader system. Either that can be a fluorescence imager from Symantec, or also fluorescence plate reader can be used, like from TCAN, from Molecular Devices. This is an instrument from BMG, exactly, yeah. So different kinds of equipment can be used to run the assays and to read out the plates. So when we're setting up a screening experiment, first thing we do is a denaturation optimization, and that means that we're looking at different temperatures, usually 4 different temperatures, to obtain the best conditions for the sample preparation. Usually that's something like 70–75% for an IgG, and there's always an optimum temperature that has determined before the project, and then all the samples can be run at the same temperature. So that's the first thing that you do, and once that is done, you are assessing the effect that the supernatant has on the assay, and this is necessary to find the optimal range for the assay, which means, like, what is the final concentration range of the IgG, which is optimal, and what kind of dilution is necessary for this. And here, in this example, we used 2 glycoforms of the same IgG that had different levels of afucosylation, and we ran the assay with samples diluted in mock supernatant, and then we diluted the samples in PBS, and you can see that in both settings, the fucosylated sample shows a higher marker binding. That's what you would expect, higher than the afucosylated glycoform, which was something like 20% or so fucosylated, and the fucosylated, the full fucosylated, had 100 or close to 100% fucosylation. And what you can see is that the assay in PBS shows a much higher delta between those 2 curves, which means that the discrimination power of the assay in PBS or in supernatant diluted in PBS is a lot higher. So as a rule of thumb, you would say that the more you can dilute the samples in a buffer like PBS, the better it will be for the discrimination power of the assay. So that's the one thing that you can derive from such an experiment. Second thing is that you can derive the IgG concentration range, which is optimal for such an assay. And you would say that here, anything from maybe 50 to 100, maybe 150 is a good range for this assay, in which we will have a real good separation because between fucosylated and afucosylated samples. So when we use that, in a screening scenario, and this is an example from screening on the 24 deep well stage. What we do is we have the samples, we know their concentration, and all the samples are diluted by the same factor and buffer. And then we perform the assay, and what we get is the fucose marker binding in percent. And at the same time, you have the titer information for all the samples. And in order to find now the degree of fucosylation, you do a normalization for titer. So now the higher bars, the high bars show you the highly fucosylated samples, and of course then the low ones, the samples with a low degree of fucosylation. And as a next step, you would do a ranking for the fucosylation, which allows you then to select the clones which are in the range that you would like to have in the clones that you are selecting and moving on to the next stage. So of course it would be good, or if one had at least one reference sample in that experiment with a known degree of afucosylation, for example this one, and then you would know that every clone or sample in the vicinity would be at a rather similar level of fucosylation. And of course, if you had more than that one reference sample, you could really have more kind of landmarks in this graph to have some kind of calibration if necessary. Okay, and that's what I wanted to show you. I hope I could show you that we have this PA201 assay, which allows you to do very fast afucosylation screenings, which means that hundreds of clones can be measured on a day, and you would have to plan, I would say, maybe a little more than half a day to do the setup experiments, this denaturation optimization step, and this dilution protocol development, but then you're ready to go. These assays work on standard equipment in the lab, plate readers, shakers, imagers, etc. So they can really be nicely implemented in CLD labs and hopefully accelerate the clone selection and finally the projects. So thank you very much for your attention, and I'm happy to answer any questions.